Welcome to this week's lab video for Biology 130A. This week we will be doing an advanced inheritance lab. To participate in this week's lab, complete the online video that you're watching and fill out the worksheet and bring that completed worksheet to lab. Also, you might want to bring a laptop computer again this week to enhance your online experience. The objectives for this week are going to be to try to understand more complicated inheritance patterns that we've talked about in lecture, such as lethal alleles, X-linked loci, which we've met previously, but we're going to be doing them in a little more complicated way. Epistasis, both dominant and recessive epistasis. Physical linkage, and then we can use the results of physical linkage to do some genetic mapping. And then finally, we'll do some pedigree analysis, and we'll make sure we can tell the difference between a pedigree that is showing an autosomal dominant trait versus one that shows an autosomal recessive trait versus one that shows an X-linked dominant trait and one that shows an X-linked recessive trait. We'll compare all of these and understand how to tell them apart. So once again this week, we will be using the virtual Drosophila lab online, and we'll do a little bit of work initially to help us recall how to use the online Drosophila lab. Now recall last week we did some simple monohybrid crosses, and we learned some things, for example, like that a Mendelian F1 crossed with an F1 will give you a 3:1 phenotype ratio, and we know that means a 1:2:1 genotype ratio. And we also did some sex-linked traits in which we saw that the patterns are going to be different between the genders, and that would tell us that this trait was going to be located on the X chromosome. This week, though, we're going to add in a little more complicated cross, in which we're going to examine lethal alleles. Lethal alleles, recall from lecture, occur when the homozygous condition prevents completion of development and lethality. Often, we find that these lethal alleles are associated with a trait that is dominant for some other condition, and that when you cross two heterozygotes, it shows a 2:1 phenotype ratio rather than a 1:2:1 phenotype ratio. This is how we detect a lethal allele. Typically, the lethality condition is considered to be recessive with respect to lethality, even though it might be dominant with respect to some other trait. To illustrate this, let's review what we learned in lecture when we compared yellow mice that have a lethal gene. In this case, we noted that when you mated two heterozygous yellow mice together, in this case we're going to indicate their conditions by big Y and little y, that they would typically produce 2/3 yellow mice and 1/3 non-yellow mice of some kind, and that this was simply the result of the fact that the yellow mice were all heterozygotes, and that the homozygous dominant mice simply didn't complete development, so we couldn't get the 1:2:1 ratio, and instead got the 2:1 ratio. So let's take a quick look at the online virtual Drosophila lab and remind ourselves how to make F2 offspring, and then as we're doing that, we might as well take a quick look at a lethal allele. So here we are back at the Drosophila online virtual lab. And when I load this up, the first thing I'm going to do is go to my options, and I'm going to try to get rid of some of these sounds and transitions. That way I can go back and I can order my flies. So I've ordered my wild type female, and now I'm going to go in and order a curly winged male. I'll add that to my cart and bring it home, check out, unpack it. Notice how much faster and easier it is with the transitions off, and I can sort my flies. One of the things I'm noticing here is that unlike the typical F1 where I get either wild type or curly winged, I'm getting both wild types and curly winged, and the pattern is the same between the sexes, so I know it's not sex linked. So in this case we said we always want to mate the mutant, so I click on the mutant for the male, and I add it to the mating jar, zoom back out, click, oh that was the female, now I click on the mutant for the male, add that to the mating jar, and I zoom back out and I go to the lab, and there's my mating jar, and I've got a curly winged female to mate with a curly winged male. And now I'll be able to go in and look at the mating and sort the flies and figure this out later. So then we will go on to examine more complex inheritance patterns using dihybrid crosses. Recall that we did some dihybrid crosses last week, and that's where we used two traits at a time. For example, last week we did shaven bristles and purple eyes as a combination, and we found that in the F2 we got the 9:3 to 3:1 ratio. Note that this ratio is what we expect when there is independent assortment, as Mendel had found. Independent assortment means that the two traits, the genes for the two traits, are located on different autosomal chromosomes. And in fact, if we were to look at a map of the Drosophila chromosome, we would find, in fact, that the purple eyes are on chromosome 2, and the shaven bristles gene is on chromosome 4, on different chromosomes and therefore sorting independently. Now, let's take a look at some of the other inheritance patterns that we might see through a dihybrid cross. So first, we might see sex-linked genes. We saw this with the monohybrid cross, but we might also see them with the dihybrid cross. Now, sex-linked loci, recall, are those that are going to be on the X chromosome. Here's an example of a variety of different traits that are located on the genes for which are located on the X chromosome. And that we'll notice them in their pattern of inheritance because we'll see a difference in the inheritance patterns between the sexes, and that will tell us we have sex-linked loci. Another type of inheritance pattern that we might see is epistasis. And recall that in epistasis, that's when you have the variation at one locus masking or blocking the expression of the variation at another locus. And for this, we're going to examine the F2 generation from a dihybrid cross. And what we would expect is that for dominant epistasis, where a dominant allele is blocking the expression of another trait, we'll detect that by a 12:3:1 phenotypic ratio in the F2 generation. Whereas for recessive epistasis, where a recessive combination of alleles is blocking the expression of a trait from another locus, then that will be detected by a 9:3:4 phenotype ratio in the F2 generation. In both cases of epistasis, we'll only find 3 types of phenotypes rather than 4 as we typically get in independent assortment. For the final type of inheritance pattern that we'll detect with our dihybrid crosses, we'll be able to detect physical linkage. Recall that physical linkage occurs when genes are located on the same chromosome. And in this case, we'll be able to detect physical linkage when we see that there are more parental phenotypes than recombinant phenotypes. So just to quickly review, let's say for example, we're looking in the F2 generation. So we take our parental cross. In this case, we have wild-type fly combined with a double mutant black-bodied vestigial winged fly. And when they mate, they produce the F1s that are wild-type heterozygotes. If we were to mate the F1s together, we would produce a series of offspring, and we get 4 different categories of phenotypes. But what we would notice is that we would have not a 9:3:1 ratio, rather we would have a case where there were many versions of the wild type and vestigial winged forms, and very few versions of what are known as the recombinant forms. These are parental offspring because they look just like the parents that started out, and these are recombinant offspring because they have combinations of phenotypes that are different from what the parents looked like when we started out the crosses. So to summarize the various types of inheritance patterns we might see with our dihybrid crosses, you might want to pause the video for a second and just review and fill out your worksheet all of the ways to detect each of the different inheritance patterns listed in the table. So we can use the results of physical linkage then to do some genetic mapping. In this case, we can do genetic mapping by using test crosses of the F1 back against recessive homozygotes to calculate recombination frequencies for 2 physically linked loci. So for example, in the case where we had the F1s for the black and vestigial winged mutant versus the wild type mutant, the F1s could be back crossed against the double homozygous recessive mutant and produce some offspring, and we would get again 4 different categories of combinations of phenotypes. And we would get those that look like the parents and those that did not look like the parents, which were recombinants. We could use these recombinants then to calculate something called the recombination frequency. And the recombination frequency is going to provide us a map distance between the 2 loci we're comparing. The way we calculate the recombination frequency, you'll recall from lecture, is to simply add up the total number of recombinants, divide them by the total number of offspring, and multiply that by 100 to get a recombination percentage. Now, in order to examine recombination frequencies, we're going to have to remember how to do a test cross in our virtual Drosophila lab. So let's take a quick look at how to do the test cross so we can remember when we get to lab. So here we are back in the virtual lab, and we're looking at our cross of our female wild type with the male sepia dumpy winged combination. And if I take those out and put them under the scope, we sorted them out and we had our two piles of F1s, our F1 females and our F1 males. To do the test cross, I'm going to take my F1 females, I'm going to put them in a new mating, I'm going to zoom out and I'm going to return to the lab. And here's a mating jar, but notice there's only females in there. In order to do a test cross, I'm going to have to go in and get my males that I originally ordered, and notice that they're both homozygous recessive for sepia and dumpy wings. So I'm going to click on that jar, I'm going to add them to the mating jar. Now there's my back cross, my F1 female back crossed with my male parental form, that's the double homozygote. So I'll put them in the incubator and I'll take them out, and now I can sort them and look at their test cross, and I can send this data to the computer to evaluate perhaps whether it's a 1 to 1 to 1 to 1 ratio, although those piles look pretty similar in size. Now because those piles looked somewhat similar, they probably are in a 1 to 1 to 1 to 1 ratio, which means that dumpy wings and sepia eyes are probably not linked. However, for genes that are linked, we can begin to calculate recombination frequencies, and then we can go on and begin to do some genetic mapping. So now that we can do our test cross and identify physical linkage and get our recombination frequencies, we can apply this to mapping multiple loci. So if we conduct our pairwise test crosses and get our map distances between each pair of genes, and remember that 1% of recombination frequency is going to equal 1 map unit, so we'll be able to measure these in map units. And let's say we have traits A and B, and we find that we have a distance of 25 map units, and we might have another comparison that we did between traits A and C, and we found that their distance was about 16 map units, and we might do a third comparison, this time of B and C, and we discovered through their physical linkage that they were separated by about 38 map units, or had 38% recombination frequency. What we can do then is we can take that type of information and we can reconstruct a logical pattern of gene order. So for example, if we have our chromosome and there's these 3 traits or genes for these traits on there, that we know that the traits that must be furthest apart are going to be traits B and C with a distance of estimated at about 38 map units. Therefore, A must be in the middle of those two. They have shorter distances between A and C and between A and B, and that this makes a logical sense, and we can get the relative order of these 3 traits, or the genes for these 3 traits. Now the other thing we're going to remember is that when we have a distance that's further apart, that map distance is going to tend to underestimate the true map distance. So if we add 16 and 25, we're going to get a larger number than 38, because there's going to be the opportunity potentially for double crossovers to occur between B and C and cancel out, and we're going to underestimate some of the recombination that's going to really have taken place. So in the final part of the lab, we're going to do some pedigree analysis. And in this case, we're going to want to be able to look at pedigrees and determine what the inheritance pattern is for each of those pedigrees. So in the case of an autosomal dominant trait, we'll look at our pedigree here, and in the pedigree we'll be able to read it by squares being males and circles being females, and those individuals that have the trait or express the trait will be individuals that will be filled in, and those individuals that are hollow do not have the trait. So in the case of the autosomal dominant trait, we'll notice that this trait does not skip generations. So you don't have a case where parents without the trait give rise to an offspring with the trait. There's always a parent that has a trait when an offspring has a trait. Secondly, we'll notice that in the case of the autosomal condition, that fathers with the trait can give to both daughters and sons. And when you see that, that tells you that this is not an X-linked condition. Finally, if we take a look at this special case here in red, we'll notice that when you have 2 individuals that both have the trait, that they can produce an individual that does not have the trait. And that would not be the case for a recessive condition. So this definitely shows you that this is an autosomal dominant trait when you have 2 individuals that have the trait giving rise to an individual that does not have the trait. And this is done because they're 2 heterozygotes that give rise to the homozygous recessive condition, which does not have the trait. We can also identify autosomal recessive traits from pedigrees, and in this case we're going to look for clues such as being able to skip a generation. That's a clue that it's a recessive trait, and a clue that this is an autosomal trait is that parents that do not have the trait can mate and produce both sons and daughters. So for example, this pair, that does not have the trait will give rise to two sons that do have the trait, and that's because they're heterozygous carriers and they can produce an homozygous recessive offspring. This couple, on the other hand, was also, both of them did not express the trait, but they gave rise to a daughter that had the trait, and that's because they also were carriers, but in this case they gave rise to a daughter that was a homozygous recessive. We can also look at X-linked dominant traits in a pedigree, and in this case we'll note that females that carry the trait will carry at least one copy of the dominant allele that causes that condition to occur, and that females that have this trait have the ability to give that condition to both sons and daughters. Conversely, when a male has the trait and it's carrying the dominant allele that causes that condition, that fathers with that trait can only pass their traits on to daughters, and that's because they only give their sons the Y chromosome. So the sons cannot receive that trait from their father. And finally, for the X-linked recessive trait, when we look at those pedigrees, we'll notice that the trait is going to show up more commonly in males than in females. Also, if a mother has the trait, then all of her sons will get that trait. Here's a mother has the trait, and her sons will get the trait, and there will be all of them. Finally, if you have a generation that's been skipped, so for example these two individuals do not appear to express the trait, and yet they give the trait to the next generation, so we know it's recessive. If it's X-linked recessive, they will only be able to give that trait to males. They will not be able to give it to females. So that brings us to the end of this week's video for the pre-lab video. So be sure to bring your completed worksheet to lab, and also we recommend bringing a laptop computer. And we look forward to seeing you in lab this week. See you then.